This is your introduction to the structure of DNA. Why is this important? Do you want to know how mutations happen, how we inherit our traits, and how proteins are made? Well, we need to understand the basics first before we can move on to these topics. DNA is one of the macromolecules we'll be concerned about in this class. DNA is known as a polymer. A polymer is a bunch of repeating monomers linked together to form a large molecule. What are monomers? Monomers are single parts that when joined together form polymers. The picture below is an analogy showing that the whole train would equal polymer because it's a large molecule. However, these single cars would represent the monomer or a single part of the whole entire polymer. DNA is composed of monomers called nucleotides. The three parts of a nucleotide are the phosphate group, the deoxyribose sugar that gives DNA its name, and nitrogenous base or nitrogen base. Now when you join these nucleotides together, you can form long chains of DNA. DNA, however, has a base pairing rule system to make sure that we can make exact copies of the DNA when it needs to be replicated. The way that these work is you have adenine and thymine. They're like best friends and they never separate. If you see one, you'll see the other. The other base pair is when cytosine pairs with guanine. And just like adenine and thymine, they are always together. A great way to go and remember this is that at Garden City, like you see below, will help you remember that A and T go together and G and C go together. The previous slide did show the base pairing rules, however it did not show the overall shape that we normally see DNA in. The DNA is known as the double helix because it looks like a twisted ladder with a sugar phosphate backbone that's on the outside edge of the molecule. The DNA is a double strand molecule. In the middle, between say the A and the T or the G and the C, it is held together by a hydrogen bond. And these hydrogen bonds are very weak and easily separated. Lastly, the sequence of our A, T, G, and C is what gives us our genetic code and makes us all just a little bit different. On this slide here, you will see the pieces that you will use to make your DNA model. Now you can choose either the yellow deoxyribose sugar or the gray deoxyribose sugar, depending on what is in your box. Other than that, you need 12 of the 2 hydrogen bonds. You need 7 thymine, 7 adenine, 5 cytosine, 5 guanine, 24 phosphate groups. You will use these pieces to construct a DNA model. I understand that it will take a while. Just keep at it and understand that this is a self-discovery lab. This file will be found on the classroom stream underneath the master objective. What you are to do with this is to place your picture of your DNA constructed model in the box. You will then use Notability to go and label each piece very specifically as to what piece you are talking about. Once this is done, please turn it into the Google Classroom turn-in spot. If you have questions, please come and see me before class on Monday so we can get those cleared up so we can move on.